Hi everyone, welcome back to my channel. And as I said in my previous video, that there are I have made so many mistakes in last one video because that was my first video and I was very nervous to make it. But yeah, let me correct those information. So there are not 17 days gap, there are 14 days gap in my biology exam. Okay, and also there are one chapter is cut it from the syllabus, so there are now 13 chapters total. Okay, so today is 13 March, and I'm going to revise the hard chapter first because see, my strategy is going to be I have distributed my biology all chapters like hard, medium, easy. Easy, I know very much, like easy or easy, like we can do if we are even lazy, so we can do easily. But like difficult chapters doing first is like a different task. It's better to understand, yeah. So I have done today principle of inheritance revision. I have made some notes of my previous one only, some are previous, and today also I have made some new. And ncrt I have, and I have this book also from pw, but these are like notes only. And like with me it's happening like till then time I'm not making my own notes, I'm not getting the point, I'm not getting the ncrt. So I have not touched the ncrt. I'll read with you all, and also I'll revise. Okay, so without any delay, let's start. Okay, so yeah, प्रिंसिपल ऑफ इनहेरिटेंस एंड वेरिएशन, ठीक है। इसमें सबसे पहले in the, and I'm going to talk like I know Hindi, I can like make people understand in Hindi, but I want to check, mm, may I taught you or make anybody understand in English. So this video is going to be full in English language. Okay, so let's start. So before going to chapter, we should know some terms like genetics, this unit, molecular inheritance, and principle of inheritance of variation. These both chapters are coming in genetics. So genetics, it deals with inheritance and variation of characters from parents to offspring. So genetic is a term which defines like the variation or inheritance. See, word inheritance, inherit, that we have inherited from our parents, progeny. Okay, and so the variation, inheritance that we have, um, that are transmitted from one progeny to another is called genetics. Okay, now inheritance what, जब कोई when the characters are passed, like when genes are passed and genes are having some character, so when character is passed from one progeny to another is called inheritance. Now second is variation. Variation means like variation, different types, like this is this, so this, like, so these both are varieties, like variation. So degree of which progeny differ from their parents, like parents, everyone in this universe are different. Why? Because they follow the term, the process of variation, because they are different from their parents. Okay, now Mendel you have heard in class 10th also. So Mendel in class 10th, like we have studies variation one chapter in biology. So Mendel is the father of genetics. Yeah. So his full name is Gregor Mendel. Like its pronunciation in my ma'am says Grigor or Grigor, like any you call anything, but we should know the name, like Gregor Mendel, or he is called father of genetics. Okay, why he is called father of genetics, will read front. But let me tell you, because he have experimented so many things and he have given many experiment for this chapter, like principal genetics. Yeah, so many experiment he have done, so he should call like father of genetics. So now there was a hybridization experiment. on garden pea. Like we are calling it in India pea, and any other country like we are calling any other with any other name. But in scientific name of garden pea is Pisum stivum. And see, there is a new way to write a scientific name. P capital ISUM space SAT, S small should be there, okay? SATVUM and underline full. No different full. Okay, this is name. It's coming in paper like for 1 marks in MCQ, like the correct way of garden pea scientific name. So yeah. So he have experimented for 7 years, 1856 to 63. Okay, and for what, like he have done artificial pollination, cross pollination experiments using several true breeding pea lines. ठीक है, ट्रू ब्रीडिंग पी लाइन्स का मतलब means like undergoes continuous self pollination. Like this is a plant. If we see, these are two plants. If we do cross pollination, means they are being in both the flowers. Yeah, different flowers of different plants. But for self pollination, a single plant and single plant, same flower, same process is done in the one single flower only, so that is called self pollination. So true breeding line is like if the plant is tall, so tall, with tall only it will be like crossed or pollinated. Okay, so that is termed as self pollination. Now जो for like what are the significance of like true breeding line, what is happening, why we should use like true breeding line. So this is continuous self pollination, we know that. Okay, this shows stable trait inheritance. Okay, this shows stable inheritance. How? Like stable inheritance means, see, if tall was the parent, so tall will be the future progeny. If tall was the future progeny, so tall will be its future progeny. So all will be true, na? True breeding line में. If we want like, if the question comes like if we want only tall in every progeny, so what should we do? So we should use true breeding line. Okay. Now express for several generation. It's like several generation continue करता है ये. He worked with 7 characters of pea plant. Yeah, so it's understood. Selected 14 tree breeding pea plants varieties. So he selected 14 true breeding lines. 14 times the same thing. Yeah. So 7 characters and contrasting traits which he have used was stem height, flower color, flower position, pod color, seed color. So so see, stem height, dominant and recessive. I'm going to tell first dominant and then recessive. First is tall and recessive is dwarf in stem height. Flower color violet and white. For flower position, axial is dominant and terminal is recessive. Okay, pod shape inflated, constricted. Pod color green, yellow. Seed shape round, wrinkled. Seed color yellow, green. Inheritance of one gene. Monohybrid cross. Okay, inheritance of one gene. Like we are now in our next topic. Okay, these are like basic MCQ is coming from which we have you read till now. Okay, now we are like coming to our next topic, which is like very important. 2 to 3 marks questions are coming from this, and even case based studies coming. So like we should like read it properly, na? So inheritance of one gene is also known as monohybrid cross. ठीक है? Mendel crossed tall and dwarf pea plant. In inheritance of one gene में he have used only one contrasting trait, that is dominant and recessive. Dominant is tall and recessive is dwarf. So when we cross both of them, yeah, so what we get? F1 progeny. seed colors will generate, plant will generate. Yeah, so all F1 are tall, no dwarf. Similar observation for other traits. Okay, F1 resembles क्या करेंगे, one parent and no trait of other parent. सेल्फ पोलिनेशन क्या करेंगे, F1 से F2 जनरेशन में प्रोपोर्शन होता है. ड्वार्फ क्या होता है, 1/4 ड्वार्फ शो होगा क्योंकि वो डोमिनेंट है. एंड टोल की वो रिसेसिव है, सॉरी, ड्वार्फ इज रिसेसिव, सो आउट ऑफ 4 it will show only one character, but tall is dominant, it will show 3 characters out of 4. Yeah, so character not present in F1 expressed in F2, those are not expressed in first generation, they are see disturbance, but we should focus on our studies. Yeah, so the which are character which are not present in F1 generation, they are present in F2 generation. Okay, now in this inheritance of gene, no blending is shown. Blending means like no high, like no blending means no mixture, either tall will form or either dwarf will form. Okay, no like medium height. Okay, no blending. So like many times I have seen this question ki in which type of like क्रॉस ब्लेंडिंग इज नॉट seen, so that is monohybrid cross, मतलब कि inheritance in one gene. Yeah, so Mendel proposed, what Mendel proposed, something was being stably passed down uncharged from parent to offspring, he called factor. Okay, so what he means proposed was कि something is that which is transferred from parent to like offspring. So something is there means something he named factor. And let me tell you, these factors are only known as gene, but it was discovered later. So here, according to chapter, he described it as factor. Okay, so now called, see it's written now called as gene, unit of inheritance. So unit of inheritance is gene. Gene code for a pair of contrasting traits known as alleles. So in gene, like this is gene. So there are like levels. This is dark blue, this is light, this then again this is ink, then this is again dark. Yeah. So these are traits, these are characters, yeah, which are transferred. So what we call this full is gene. Okay. Now gene, one gene, this is called chromosome. Now we have taken only one gene, single gene. Now these are called alleles, which have traits. Okay, traits are like eye color, eye shape, eyebrows, hair color. Yeah, these are traits, characters basically. So if in paper it's came, how we are going to explain it, and it's very important. Sometimes it's given like it's most important, monohybrid cross. So let me take a notebook, you also please take a notebook or a page. Okay, and start writing it. Yeah, I'm taking this notebook. I'm sorry. So like monohybrid cross mein what we are going to take, see monohybrid cross, first write. Okay, if we take both capital T, TT, that means tall. Capital T shows that this tall is dominant over dwarf. Recessive. So recessive is small t, and this is dwarf. Understood? Understood? Now if we'll cross this TT and TT in F1 generation, we'll get all TTs, capital T, small t, capital T, small t. All the uh, offspring are going to be same, all are tall. Okay, but in F2 generation, when we cross TT, capital T small t, capital T small t, what we are going to see, like capital T, capital T, capital T small t, capital T small t, and small t. So we got 3 tall and one completely dwarf. Now see what are important in this. There is a like if anybody is asking like question, so he will ask from phenotypic ratio and genotypic ratio. Yeah, like see, phenotypic ratio means pheno, which we are uh, physical appearance mainly. Like if I am saying how it's like, if anybody will see me, what he will see, he will not see she's a very good girl, she's this, no. What they will see, her hair color is this, her hands have 5 fingers. So what is outer appearance, that is phenotypic. And genotypic, what are inside me. Yeah, genotypic like, mm, she's a good girl, she's like good in teaching, she is like my, like what we are saying that, which is not seen physically. Okay, so that is genotypic ratio. So if we see a plant, what we can see, it's uh, its flower are pink, its height, its seed color, its leaf color, yeah, we can see it, its stem color. Yeah, so that is only. But genotypic, how it is, yeah, genotypic like, uh, whether it's homozygous, it's heterozygous. Homozygous means both are same, capital T, small t, homozygous, both the traits are of same, like, yeah. If it's heterozygous, so what we are going to do is capital T small t, capital T small t. Yeah, so same, that is heterozygous. So in this, what we are going to do, phenotypic ratio and genotypic ratio are same in F1 generation. Okay, but it's different in F2 generation. How? Like 3 is to 1, it's phenotypic ratio, but 1 is to 2 is to 1, it's genotypic ratio. Understood? Like we are doing now revision. If you want to like start from starting from zero, you are just appearing, you are just coming to 12th, I'll make any other series for that. But this is like a revision series, we have already studied it first, so this is also named as revision series. Yeah, so as a word, I'm going to just revise. Yeah, and like, who is not able to like revise properly, he can sit with me and he can revise, open books and just revise. Okay, so for them I'm making this video, and for me, how I'm like, I want to also check that how can I like make people understand in English language. Yeah, so just for that. So now the question, now the next topic is test cross. Monohybrid cross we have done. See what we have read till now. We have read genetics. What is genetics? भाई कुछ something is passed from parents to offspring. Inheritance. Character is passed from one parents to progeny. Progeny to offspring. Yeah, and variation or different. What makes variation is something that makes parents differ from their offspring. Yeah, and then Mendel laws of inheritance. We have read something. There was a Gregor Mendel. Yeah, and 1856 to 1866. 7 characters, 14 traits, contrasting traits, characters, um, flower color, uh, stem height, pod color, pod shape, seed color, seed shape, बट कुछ तो भूल रहे हैं, कुछ याद करो. Flower position, flower position. Yeah, then we have read inheritance of one gene, that is called monohybrid cross. Monohybrid cross we have read dwarf, 1 by 4, 1 by 3 by 4, no blending. Yeah, F1 generation only tall and dwarf. F1 all are like cross pollination, but in F2 when we do self pollination. Phenotypic, genotypic, 3 is to 1, 1 is to 2 is to 1. Then now next is test cross. When F2 hybrid is crossed with recessive plant. See, now what we were doing, we are taking tall and dwarf plant and then we were getting F1 generation. But now we have taken plant F1 generation, self pollinated F2 generation. Now this F2 generation we will cross with any other plant. Then it is said to be test cross. This we have understood with the determine whether. So why we are doing test cross? It's monohybrid cross, either dihybrid cross. Why we are doing test cross? As a name, test cross means something we are testing for anything. So test cross is used to determine whether dominant character is coming or the recessive character is coming, whether the plant is going to be going to be, yeah, no, what is going to come, that we are not, like I am there now, I want to check whether my mother or father was homozygous or heterozygous, whether they were dominant or recessive. So test cross is that used to determine the homozygous or heterozygous dominant progeny. Yeah? So determine whether dominant character is coming from homozygous dominant genotype or heterozygous genotype. Okay. TT, if capital T crossed with small t, okay, homozygous tall with homozygous dwarf, then we get only tall. Yeah, that we know. If heterozygous is crossed with homozygous dwarf, see, homozygous dwarf is like permanent, okay, but the first one, capital T, homozygous. देखो, now I can understand so many of you are like confused, but when you will read your book, it's also in your book, I'll make sure I'll go through it also. Okay, as we complete our notes, I'll go through from this. Okay, I know, just try to understand. Homozygous tall means, homozygous means both the traits are same, so capital T, capital G. Heterozygous dwarf, heterozygous dwarf, heterozygous heterozygous dwarf, so it cannot be happen, it can't. Yeah, so heterozygous tall means capital T, small t, understood. And dwarf can only be homozygous, no, small t, small t, small t, small t. Okay, so we will get, for heterozygous we'll get two outcomes. One is like heterozygous tall and small also, both dwarf also will get, so both tall and dwarf. If all tall progeny in test cross is there, so tallness coming from tt. And if both tall and dwarf progeny tallness is coming from capital T and small t. So this is like only testing for whether the parents are heterozygous dominant or homozygous dominant, genotypic. Okay, now next is law of inheritance. We have read monohybrid cross. We have read, what we have read? Tell me. Have you forgot? No. We have read test cross for testing. Now it's law of inheritance. What is the? There are like two laws of inheritance. Let me read. One first is how the like characters are passed from one progeny to another, that is called inheritance. So how, question is how, so it, because it follows two laws. For one, first one is law of dominance, like characters are controlled by discrete unit factors. Yeah, characters are controlled by gene, that I have told. Factors occur in pair, yeah, factors occur in pair, small t, capital T, like, okay. And in dissimilar pair of factors, one member of one pair dominant over another recessive. So both of those characters, one is dominant and one is recessive. Dominant means it will show its power, yeah, and recessive means it's just repressed because of dominant. Yeah, we can understand by the terms only dominant and recessive. So yeah, when the traits are going to express, so one is going to be dominant, one is going to recessive. So as the name of law of dominance means someone is going to like dominate the other one. So it explains only one parental character in monohybrid cross in f1 generation of both f2. ठीक है, see, In F1 and F2, only one character is going to express, okay, that is tall or either dwarf. Fine. So this is law of dominance. Means dominant character is going to repress the recessive character. Yeah. Like we have read in monohybrid cross only, tall and dwarf. Tall was dominant and that dwarf was recessive. In F1 generation, all were tall. Why? Because it was following law of dominance. No? Now, second is law of segregation. Segregation means separation. Yeah. So alleles do not show blending. Yeah. In monohybrid cross also they do not show blending. Okay. Parent contain two alleles. Yeah. Parent contain two alleles. Okay. During gamete formation, two pair of alleles segregate. Okay. Gamete receives only one, either homozygous parent or heterozygous parent. Okay. So what are they are saying? Ki when the genetic gamete is formed, when the gamete is formed, two alleles are separated. Okay, these got separate. So that is only the term like law of segregation. Why they are not showing blending? Monohybrid cross. Why it is not showing blending? Because it follows the law of segregation, in which during the gamete formation, both the pair of alleles got separated, and it forms either homozygous or heterozygous genotype parent. Sorry. Yeah. Same type of gamete. And if homozygous parents are formed, then same type of gametes. And if heterozygous is formed, then different types of gametes. It's understood. It's like basic for this chapter, but I am revising even basics. So next is incomplete dominance. Sorry. Next is incomplete dominance. In this, neither of the two alleles are completely dominant over other. Now see what we have read: law of inheritance, law of are described into two: law of dominance, law of segregation. Law of dominance means someone is dominating the other character. Yeah. And in segregation they show no blending, they just gamete formation ke means during gamete formation they got separated. Segregation. Now it's incomplete dominance. First he has said that law of dominance is there. Now he's saying incomplete dominance means no one is like dominating the other. Like so in this the blending can happen. Yeah. So in this neither of two alleles are completely dominant over the another. Incomplete dominance. Flower cup color in Snapdragon or Anthurium are capital R, small r, red, white. Gamete are smaller and capital R. So when they are like crossed with each other, what is formed? Pink color. Red and white, pink color. So see, it is not showing dominance. Yeah. So this is just a science we should know. So in incomplete dominance, it's a very important topic because all are thinking like law of dominance. But trust me, this is I have seen in all sample papers. Incomplete dominance. Sorry. F2 generation, when we cross pink. So what we get? Phenotypic ratio is equal to genotypic ratio. 1 is to 2 is to 1. Okay. We have read law of dominance, incomplete dominance, now co-dominance. These all three are like confusing, and that is why they are asked in paper many times. Okay. Co-dominance. F1 generation resembles both parents. In co-dominance, in F1 generation, both parents are like resembles. In this, there is an example of ABO blood grouping in human being type of red blood cells. Okay, so ABO blood group are controlled by gene 1. Okay, and plasma membrane of RBC has sugar polymer that protrude from its surface. So like this is RBC. We have taken example of grouping, like ABO grouping, AB O, B positive, A positive, B positive. So why it is so? Because it show codominance. So these are present, blood is present in RBCs. So we are taking of RBC, like this is RBC. A polymer of sugar is protrude out, means it's like out of surface, out of plasma membrane, like this. Okay, so that is known as protrude. Here I have read this word. So from its surface, okay, we have got kind of sugar is controlled by gene 1. Okay, gene sugar also, and the ABO blood group also, gene 1 is controlling both of them. Okay, and the gene 1 has 3 alleles. Gene 1 has 3 alleles, 1 2 3. First is IA, IB, and this is small I. Fine, it is recessive. So if a gene is having more than 2, then it is known as multiple allelism. Okay, these are allele now. So these are multiple allelism because they are more than 2. IA and IB produce slightly different form of sugar. Okay, I does not produce any sugar. Recessive do not show any sugar. Human is diploid, process 2 out of 3 genes, alleles. Okay, so as human is diploid 2n, so out of 3 we have 3 alleles, so only 2 alleles show their characters at one time. I and IB are completely dominant over I. See, this is recessive and they both are dominant over I. I and IB present together both express their own sugar. Okay, so as blood group A, B, AB, O, how these are formed? Like this question is also very asked. Like how if a case base is coming on this like topic. Like if a child, if a mother is A, father is B, what is going to be the offspring blood group? So we are using this. If the mother is homozygous homozygous A and father is A, so A is formed. And if mother is A and father is recessive, then also A. And same as for B, if both are B, then B is formed. If one mother is recessive and father is dominant, then also B. How AB is formed? When both have like A and the other one have B, so both are AB. And when O is formed, when both have recessive blood groups, then O is formed. Okay, sorry. Now, inheritance of two genes. What we have read, see, law of dominance. We have read like laws. Codominance, we have read blood groups. Okay, gene 1 is showing blood groups and sugar it's controlling, and ABO, AB, A, O. We have understood. Yeah, incomplete dominance means pink color was coming in which, so they have taken the example of snapdragon and n3 new. And law of inheritance was also something to law of dominance, someone is dominant over other. Like you have to, how you can revise, you have to go in past, you have to not touch notebook. Okay, you have to just remind in your brain what you have, like what you can remind. Okay, what you are not reminding, just read it. Okay, next is inheritance of two genes. Dihybrid cross, we consider two characters. Let me check this video is on or not. Yeah, it's working all right. Okay, so now next is inheritance of two genes. Okay, that is like inheritance of one gene was called monohybrid cross. So inheritance of two genes is called dihybrid cross. So we consider, in this we will consider two characters. In those we were just explaining only one character, but in this we are taking two characters, seed color and seed shape. Okay, yellow and round are dominant. Yellow is seed color and seed shape is round. Seed color is green and seed shape is wrinkled, means recessive. Means just ignore the voice from outside, we'll just focus on this. Yeah, so round yellow, like parents which we are taking, round yellow and green and wrinkled, means uh, dominant and recessive. So what we are getting in F1 generation, so we are getting round yellow, round green, wrinkled yellow, wrinkled green. Okay, when and then when we do cross in like F2 generation, what we will get, we will get that we got total 9, 10, total 16 outcomes, total 16 offspring we will get. Okay, in F1 generation only 4 and in F2 generation 16. So 16 are from 16, 9 are round yellow, round green are 3, wrinkled yellow are 3, and wrinkled green are 1. So their phenotypic ratio is 9 is to 3 is to 3 is to 1. And if we will talk about the genotypic ratio, so it will be very large, because only like 1 is to 2 is to 3 is to 1 is to 2 is to, like it's going to be big. So it is generally not asked in paper, but I know like one we can make easily, like round yellow 1 is to 1 is to 2 is to 1, like this. Then second is law of independent assortment. See, what we have to like what we have to learn from this chapter is about laws. Laws are like bhai gem for this chapter. Okay, so now again one is law. So law of independent assortment, it states that when two pairs of trait, okay, are combined in hybrid, one pair of character segregate independent of other pair of characters. Okay, what it is saying, tell me, tell me. Okay, okay, okay. F1 generation RY. Independent assortment. So let me like this is, I'm also not getting it. So we are like revising it together. So let us revise again. It states that when two pairs of traits, like this is, these were alleles, this is gene. Gene is carrying some traits. So when two pairs of trait, okay, are combined in hybrid, okay, one pair of character, okay, segregate independently of other pairs of characters. Okay, what it is saying, when we hybrid two pairs traits, when we are hybriding traits of two pairs, two characters, then one character is independent of other characters. How? Like if in dihybrid cross we can take example, if we are taking two characters, seed color and seed shape, then four outcomes were there in F1 generation and 16 outcomes were there in F2 generation. So in F1 generation, which are like four characters, they will show 25%, 25%, 25%, 25%. Okay, all are like independent, all follows independent assortment, because they are independent of each other, because they are, this is independent from this is. So what I found are round yellow, round green, wrinkled yellow, wrinkled green. Okay, so these produce 25% of total gamete. Okay, this is only known as independent assortment. Okay. Now this rediscovery of Mendel's work, now after so many years, now there was rediscovery, someone rediscovered of the work. So Mendel's theory was remained unrecognized till 1900. 1856 to 1863 he have experimented all this. 60 after 363 1863, no one had worked on this, but again after 1900 it was like rediscovered. Okay, it was like remain in talks and all things. So why rediscovery was there? Because first there was lack of communication. Okay, communication, like he was like giving all its theory in English, but the scientists were uh, not understanding those his words, scientific names. Like he have used in first P, but when he used Pisum stivum, all scientists were okay with that. Yeah. So lack of communication. His concept of genes, stable, discrete unit alleles do not blend. Okay, it was not accepted by continuous variation in nature. Okay, using mathematics to explain biological phenomena, he used mathematic expression. So like in when I was in class 10th, let me tell you, our teacher tells to remember this ki we have taken uh, biology because we don't like maths, and he have only included maths in biology, so there is no existence of that. So this is a way to learn this. So using mathematics to explain biology phenomena, he could not provide only physical proof for existence of factors. Okay, he have only given like physical proofs, no like grammatic proof, no like scientific proof. What he have seen, that was physical, and he have just given those. So these were like limitations of its work. So now after 1900, three scientists came and they rediscovered Mendel's work. Okay, so what, who were those three scientists? D Veris, DEVRIES, D Rice, D Rice, Corven, Cor Corven, Corvens, Von Teschmark, Von Teschmark, VonTeschmark, Von Teschmark. So he rediscovered Mendel's work. Okay, these three. What were there? De Vries, okay, Corven, okay, and third was Jones something. Von Teschmark, DCV, DCVT. So D means D price, DC Corven, V Von, T means Teschmark. Okay, till now we have read. Okay, now it is like chromosomal theory of inheritance. Now if the chapter is genetics and chromosome didn't came, it just feel like why. So as weight is like over, so chromosomal theory of inheritance, what it says. So there were two scientists in 1902. What they discovered, they like observed the behavior of chromosomes in 1902. Who were those two scientists? Walter Sutton, Walter Sutton, WALTER Walter Sutton, and Theodore Boveri. Theodore Boveri. Theodore, Theodore Boveri. Okay, in 1902. Okay. The behavior of chromosome was parallel to the behavior of gene and used chromosomal movement to explain Mendel's laws. In diploid cell, so yeah, where I was, like my mother came in this room, so I will just open the door. Just don't get mind, I'll be there. So I was saying that the behavior of chromosome was parallel to the behavior of gene. Okay, as chromosome behave, same the type gene is like working, and used chromosomal movement to explain. Okay, he have used chromosomal movement to like explain Mendel's law. So in diploid cell, in 2n cell, chromosome occur in homologous pair gene. Okay, gene also occur in allylic pair. So in diploid cell, like in humans, chromosome occur in homologous pair. Means single chromosome cannot be there. Homologous means crows like this, homologous. Okay, homologous means same type, this is blue, this is blue, same and joined together, so this is homologous pair. This and one more, same homologous pair. Heterozygous, blue cross and one is black cross, so that are heterozygous pair. Okay, gene also, and in say similar way, gene also occur in allylic pair. So the two allele of gene pair are located on homologous sites on homologous chromosome. Pairing and separation of pair of chromosome would lead to segregation of pair of factor. Okay, knowledge of chromosomal segregation with Mendelian principle is called chromosomal theory of inheritance. Okay, so pairing and separation of pair या फिर separation of pair of chromosome would lead to segregation of pair of factor. Knowledge of chromosomal segregation with Mendelian principle is called chromosomal theory of inheritance. Experimental verification of chromosomal theory of inheritance. Thomas Hunt Morgan and his colleagues, what they have worked on: Drosophila melanogaster, fruit and flies. Okay, so they could be grown on simple synthetic medium in laboratory. Okay, excuse me. So yeah, यहाँ वॉशरूम अटैच्ड है ना, तो यहाँ पे आते जाते रहते हैं, but we'll just focus on this. Sorry for that. Okay, so we were like experimental verification of chromosomal theory. So what was chromosomal theory? I have not read this page and I'm not getting it also. So chromosomal theory of inheritance. In 1902, there were two scientists. We are just doing it. There were two scientists, Walter Sutton and Bovgeri. No, Theodore Boveri. Okay, in 1902, they have explained the law of inheritance by chromosomal theory. So, as the show chromosomal pair shows in homozygous uh, pair. Okay, similarly, gene also show alleles in pair. Yeah, and this was like in diploid cell, chromosome occur in homozygous pair and gene also occur in allylic pair. Yeah, as the same. And as the we have read law of segregation, similarly, law of segregation is given by chromosomal theory of inheritance of pair, like would lead to segregation of pair of factors. So they would, if chromosome is separating, then the gene is also separating. So with the, like this is chromosome gene is inside chromosome na. So if chromosome is like separating, then gene will also separate. Okay, knowledge of chromosomal segregation with Mendelian principle is called chromosomal theory of inheritance. Experimental verification of chromosomal theory of inheritance. Okay, Thomas Hunt Morgan and his colleagues, Drosophila melanogaster, fruit flies. Now, when they have given experimental verification of chromosomal theory, so what they have said, they have taken Drosophila melanogaster, which is also known as fruit flies. It's a name of plant basically. Okay, so why they have used this? Because they could be grown on simple synthetic medium in laboratory. So these plants can also be grown in laboratory. Okay, and they complete their life cycle in 2 weeks. And they complete their cycle in 2 weeks. If they'll take month and year, so it is difficult for like experiments now for their outcomes. So because of this, like it takes 2 weeks, this plant was used, and a single mating produce a large number of progeny flies. If we do single mating, single like inheritance, so they give variation of progenies, variation in offspring. Fourth is there was clear differentiation of the sense of male and female flies are easily distinguishable. Okay, so the male and female flies are easily distinguishable. Yeah, heredity variation that can be seen with a low power microscope. So heredity variation can also be seen from low power microscope. Linkage and recombination. So Morgan carried out several dihybrid cross in Drosophila to study that were okay, not till here, but together. Linkage and recombination is also an important topic. Okay, so we have to just see from starting. We have read principle of inheritance and variation, some of the definitions, genetics, inheritance. Okay, we have read variation. Then Mendelian laws came. George Mendel came 1956 to 1963. Okay, and then हमारे true breeding line was there. Sorry. Then characters, contrasting traits were there. 7 characters, 14 traits. Yeah. Then our monohybrid cross was there. Then how to explain it with Punnett square was there. Then test cross was there, testing. Okay. Then after test cross there was a laws of inheritance, law of dominance, law of segregation, then incomplete dominance, then codominance. Yeah. Then was our, I think, dihybrid inheritance, dihybrid cross, then independent assortment was there. Yeah. Then rediscovery of Mendel work. See, now I'm asking you a question. There were 3 scientists. Who were they? R. Nee. Corwin, von Teschmark, and one more was there. CVT. D. D. Rice. D. Rice. Okay, see, these are like when we are making this mnemonics, na? So they are like D, C, V, T, D, C, V, T, D, C, V, T. C, D होता है और T भी होता है, उनको उल्टा कर दो, D, C, V, T. So D. Rice, Corwin, and von Teschmark. Then chromosomal theory of inheritance. Okay. Then experimental view of verification of chromosomal theory. Then now it's coming linkage and recombination. Okay. So, so it's still there. Linkage and recombination. So Morgan was a scientist, carried out several dihybrid cross in Drosophila to study that were sex linked. क्या जो ये प्लांट्स हैं वो सेक्स लिंक्ड होते हैं? तो ये फाइंड आउट करने के लिए जो साइंटिस्ट थे मॉर्गन, उन्होंने एक प्लांट लिया, उन्होंने एक प्लांट को स्टडी किया जिसका नाम था डोर्सोफिला। उसमें उन्होंने बहुत सारे डाईहाइब्रिड क्रॉस करवाए, क्या पता करने के लिए, क्या वो सेक्स लिंक्ड होते हैं, ये पता करने के लिए। द क्रॉस वर सिमिलर टू डाईहाइब्रिड क्रॉस। ओके, कैरिड आउट बाय मेंडल। तो जो मेंडल, सो the crows which were like experimented by Mendel, they were same as the Morgan experiment, because he have also taken the Drosophila and Anthracium, and he have also taken Drosophila. So Morgan hybridized yellow-bodied, white-eyed female. So it is talking about humans. So what he have taken, yellow-bodied, okay, and white-eyed female, okay, and a brown-bodied and red-eyed male, and intercrossed their F1 generation. So F1 generation came, now they were crossed. Okay. He observed two genes did not segregate independently of each other. Okay. What he have observed, that there were two genes which did not segregate independently. Okay. In Mendelian, we have studied that law of independent assortment in F1 generation, that all show 25%, 25%, 25%. Yeah. But in this, two genes did not show segregate independently of each other. And F2 ratio, F2 generation ratio deviated very significantly from 9 is to 3 is to 3 is to 1 ratio. Okay. First, before it was like, let me check. Yeah, I also forget. No? Before also this was there, now also ratio. And ratio also changed. They are saying like phenotypic ratio. In F2 ratio is same, but I think phenotypic ratio is different. Yeah. Genes were located in X chromosome. Okay, X chromosome. Two genes in dihybrid cross. Two genes in dihybrid cross were situated on the same parental gene. Okay, same chromosome. Okay, same chromosome. On same chromosome, there were two genes. Okay, parental gene combination higher than non-parental gene. So, parental gene combination is higher than non-parental gene. Have you got it? Yeah. How? So they have given this, and I am telling you, I am not understood this. I am not getting this. But we should like think it off, and we should now practice so that we can perform well in our paper. So Morgan attribute this due to physical association of the two gene and coiled the term linkage. How? Morgan attributes this due to physical association of two genes, okay, and coiled that they, what Morgan say कि how the two genes are physically coiled to each other. Coiled, well, how they are physically attached to each other. This is named as linkage. Okay, they are like separated, or they are less linkage, high linkage, high linkage. So that is known as linkage. Recombination. What is recombination? Generation of non-parental gene combination. Generation of non-parental gene, okay, combination. So when non-parental gene combination is known as recombination, genes were tightly linked. When genes are tightly linked means high linkage. Tightly high linkage means less recombination. Gene were loosely linked means less linkage, high recombination. Got it? Yeah. So yeah, this is like asked in paper. This can come in case base. So Y plus and W plus are shown with Y plus is brown and W plus is red. They both are like male, and yellow is shown with Y and white is shown by W. Okay, and if the color is from brown and red, then it is said to be wild color. Wild, WILD. Okay, and in females, yellow and white, just no any other name. Okay, so in F2 generation, it's like data. Parental gene shows 98.7 recombination, and the offspring shows 1.3% recombination. Okay, now it is Alfred now, a scientist came, and what he have given, Alfred Sturtevant. Alfred Sturtevant. ALFRED. Alfred Sturtevant. He have given the frequency of recombination between gene pair on same chromosome as a measure of distance between gene. So he have given frequency ki how it is high frequency, low frequency of what, recombination between gene pair on same chromosome as a measure of distance between genes. Okay, yeah. Mapped their position of chromosomes. So what they have done now, he have also given their position. Like this is a chromosome. So where is gene present actually? Here, top, bottom, like. So they have, this is known as genetic map. Okay, and used in human. Sorry, I am just feeling cold. So used in human genome sequencing. Okay, human genome sequencing, like what is sequence, gene 1, gene 2, gene 3 is up or downward. Yeah, so that is Alfred Stoker. We have read linkage and recombination. What is linkage? We have taken yellow female, yellow white, and brown and red male. We have crossed, dihybrid cross. So F1 generation, two characters are not independently assortment, not showing, and F2 generation is significantly diverse. 9 is to 3 is to 3 is to 1, and then Morgan attribute this to the physical appearance, physical association of two genes, and they call it. He coiled the name. He coiled the term linkage. Recombination, non-parental gene combination is said to be recombination. When the linkage is high, recombination is less, and when the linkage is less, then the recombination is more. Then a scientist came, Alfred Sturt, went, okay, frequency of recombination, uh, gene mapping, okay, genetic map, and sequencing of human genome. Then next is also an important, okay, now these endpoints are the very important, these disorders, syndromes, sex determination. So sorry for all the disturbance, but let's start our topic. Polygenic inheritance. What is polygenic inheritance? So when inheritance of a character in which character is controlled by many genes, like one character first, like in normal inheritance, one character is controlled by one gene, but in this polygenic, as a name, poly means many. So as a name, inheritance of character in which character is controlled by many genes, and intensity of characters depend upon the number of dominant alleles. How many dominant alleles are there? So intensity depends on that. Okay, the genes are called polygenic genes. These genes are called polygenic genes. Okay, they have given example of human skin color. There are three genes who determine the like human skin color. First is dominant allele, who like, oh, if all are dominant, all three are dominant, so dark skin color. If all recessive, then lighter skin color. If all are like intermediate, like if all homozygous dominant, homozygous like recessive, but they are heterozygous dominant, then the intermediate skin color is formed. Okay, each type of allele is genotype would determine darkness or lightness of skin in an individual. Okay, it's very, yeah, like first I was thinking I cannot read, but now when I am like explaining you all, I'm getting the points very clearly in my mind. Okay, and now the second term is pleiotropy. Pleiotropy, yeah, see these polygenic inheritance, pleiotropy, pedigree, and these are like similar, so I was like little bit of confusing, and you may also, but eh, when you will like revise, you will write, you will read, you will clear all those points. So now next point is pleiotropy. What is pleiotropy? It is the condition in which single gene control more than one phenotype typic expression. Okay, in which single gene. In polygenic was one character was controlled by many genes. In this, single gene is controlling more than phenotypic expression, मतलब कि characters. Now, opposite of polygenic inheritance means one gene is gene. One gene is controlling many characters or phenotypic expression. Such gene is called pleiotropic gene. Okay, such gene is called, okay, example, phenylketonuria, autosomal. Example, phenylketonuria, autosomal recessive disorder. Okay, this is seen what it is. Phenylketonuria, it is a autosomal recessive disorder. I think it's a disorder. Yeah, I have read it. Yeah, phenylketonuria. Read, like read it in description front. Okay, disorder caused by, it is caused by mutation, okay, in gene that code for the enzyme, okay, phenylamine hydroxylase. Okay, phenotypic expression, mental retardation, reduction in hair, skin pigmentation. Okay, sex determination, next topic. So what we have read, linkage, but polygenic inheritance and pleiotropy, opposite of each other. Now, sex determination. It as sex determination as a name. How we can determine whether this is male or a female. Okay. So it is meant by which the distinction between male and female is established in species. Okay. Chromosomal sex determination. Now, determination through chromosomes. Okay. If XX and XY. As we know, like in humans, we have read in class 10th, class 8th, that when like female contains only XS, XX, but male contains X and Y. So female only have tendency to like form girls, but boys are like coming in offspring because of only boys, because they are having XY. If XX is formed, then a girl, and if XY is formed, then a boy is coming in the next offspring. Okay, so in this sex determination, female is homogametic. Okay, and that produce only one type of gamete. Okay, male is heterogametic, XY. Okay, and male produce two type of gametes. Yeah, we know both gametes have same number of chromosomes. Yeah, we know 46, 46. Yeah, so in Drosophila and in humans, XX and XY sex determination is followed. Mean in like female gametes, 2A XX and 2A plus XY is formed. AY in male gamete. Now second type is coming, XX female is same, but XO type. Uh, in place of Y there is O, means nothing. Recessive means zero. Female is homogametic, only one type of gamete is produced with X. Okay, male is heterogametic, two type of gamete are produced, one with X and another without X chromosome. Okay, A0. So it is seen in grasshopper. Okay, now third, it's very important and it's like assertion based, and like these questions are coming from this, because this we haven't heard in like 8th class or 10th class or in 11th class, but we'll first time we are reading it in 12th class, so it's already like important. So z z and zw type. So female is heterogametic. In this, in all of those, female was homogametic, but in this, female is heterogametic, that is Z and W. So these two types of gamete, one with Z chromosome and other with W chromosome, and male is homogametic. Okay, in this, male is homogametic, that is Z and Z, only one type of gamete with Z chromosome. So example of this is bird. Understood? Yeah. Sex determination in honeybee. Honeybee, yeah, we have read in bird, we have read in grasshopper, we have read in Drosophila human being. Now, like variation is seen in honeybee, sex determination in honeybee. Let us see. So, the sex determination which is shown by honeybee is haplodiploid. What? Haplodiploid. Okay, what is? It is on basis of number of set of chromosome an individual receive. how like how many pairs of chromosome a offspring or a progeny is receiving, it is basically depending on that, whether it is haplo, whether it is diplo. Okay, so it is known as haplodiploid sex determination. Okay, so unfertilized egg develop in male. Unfertilized eggs are formed in develop in a male, and fertilized egg develop in female. Okay, female is diploid and male is haploid. So in the same, if unfertilized is there, means that is haploid. Okay, fertilized means it can divide, so diploid. So female is diploid and male is haploid. Okay, female have 32 chromosome, and male, honeybee means female, and male means drone, that is male version of honeybee. Yeah, so 16 chromosome. I don't know how we are like distinguishing between the honeybee, like this is male or this is like female, but we are studying, so we should study now. Like in female 32 chromosome, and in male 16 chromosome. In male, mitosis means, mitosis is, what is mitosis, like this question was asked in my practicals, that what is difference between meiosis and mitosis, and I was like, what question they are asking. But see, in mitosis, same, mitosis is simple word, same chromosome, same progeny. Chromosome, the number of chromosome in the progeny is similar to the number of chromosome in offspring. But in when in female there is a like meiosis. What is different in mitosis and meiosis? In meiosis, number of chromosomes are halved in the offspring. Okay, so 30 chromosome, meiosis 16 and 16, and mitosis 16. So both from female 16 chromosome and from male 16 chromosome. Then female 32 chromosomes are formed, and when only female gametes are there, then parthenogenesis means male is formed, and that is 16 chromosome. Understood? Yeah. Parthenogenesis and fertilization. Parthenogenesis means same, no division further. But fertilization means male female fertilized. Yeah, it's easy. Now I'm feeling this very easy. Next topic is mutation. We have read linkage, recombination. Okay, Alfred Sturtevant. We have read polygenic inheritance, pleiotropy, sex determination, XXXY, XXXO, ZZZW, and oh, haplodiploid sex determination in honeybee and drone. Now we are on mutation topic. See, alteration of DNA sequence, change in genotype and phenotype is mutation. Mutation, something like, if we can, according to English, mutation means something is like mixed. Mutation means mixed. Okay, so alteration of DNA sequence. Alteration, like DNA sequence was 1, 2, 3, 4, 5, but now due to mutation there is 5, 2, 1, 3, 4. Understood? So alteration means like positioning, vary in positioning. Okay, so of DNA sequence, change in genotype. So because of sequencing alteration, there will be change in phenotype and as well as in genotype and phenotype both. So because of this, loss or gain of segment of DNA alter chromosome, like deletion or insertion of duplication of segment alter chromosome. So mutation, how it can be, like whether the five, if from five genes we can like delete one, so four chromosome mutation. If we can like insert some any other, or we can duplicate something, so the sequence will be altered. Yeah, so result in chromosomal abnormalities. Okay, so this will result in chromosomal abnormalities, because here alter is being, so like basic, it's chromosomal abnormalities will be like disorders will be. Yeah, so commonly observed as cancerous cell. Yeah, abnormalities means cancerous cell, very harmful cells. Okay, mutation due to single pair, bare pair of DNA, point mutation. Now there is two types of mutation: point mutation and frame shift mutation. Point mutation, what is saying? Mutation due to single pair. When mutation is seen in single pair, yeah, yeah, because single pair of DNA mutation is seen. Or deletion or insertion of base pair of DNA, whether we delete something or we insert something in DNA. Like in DNA, we insert one gene or we delete one gene. So that is called as frame shift. Means shifting we are doing a mutation. Now the important and like case-based topics are coming from this chapter. This topic, sorry. Pedigree, pedigree, pedigree analysis. Pedigree analysis. Pre-degree is not there. P degree is there. Analysis. Analysis of trait in several generation of family. Like to observe the traits coming in a family. Like in me, if we see my hair color, my eye color is coming from my father or mother. In those, whether they were like coming from my grandmother or grandfather. Yeah. So for analyzing of trait in several generation of family is called as pedigree analysis. A particular trait is represented in the family tree over generation. Yeah. It is a strong tool which is utilized to trace the inheritance of specific trait, abnormality or disease. From this, like pedigree analysis is used in like uh, observing the traits or disorders or abnormalities which are coming from our progeny to progeny to progeny, generation to generation, family to offspring. Yeah. So a number of disorder is associated with the inheritance of changed or altered gene or chromosome. Fine. So let me show you. Are you able to see this? This Are you able to see? Yeah. Have you seen? Now let me show. Square box is used for male. Circle is used for female. And the diamond shape, sex unspecified. And when they are filled, then that means they are affected. Okay. When male and female are linked with one line, then they are mating, then mating is done. And when double lines means mating is done between relatives. Okay. Parent above, children below. Parent above and children below. Parent. Okay. So parent above, children below. Parent with male child with disease. Okay. And when there is a 5 written in a diamond, then it is means 5 unaffected offsprings. Okay. Now pedigree analysis is very easy. Okay. But now it is easy, but when its questions are coming in case based, it's very like not difficult, but it is like it is also doable. We can do it, but it's we have to think very much on that topics, conceptual basically. The next is Mendelian disorder. What are the disorder of Mendelian? He have given like they are mainly determined by alteration or mutation in single gene. So disorder is basically discover is being because of alteration in sequence, means mutation in single gene. This disorder can be transmitted to the offspring. Mendelian disorder can be transmitted, raised in family by pedigree analysis. Dominant or recessive can be linked to autosomes and sex chromosomes. These are linked to autosomes and sex chromosomes. Okay. Now first is color blindness. Mendelian disorder, first one is color blindness. When like I'm not able to see red color, okay, or I'm not able to see white color, so that are color blindness. Linked recessive trait. When sex are linked, both sex are linked and both are recessive, then this disorder will come. Okay. defect in either red and green color cone of eye. Whether they are not able to see red, or either they are not able, either they are not able to see red or green color. Okay. Failure to distinguish between red and green color, whether this is red or green color. Yeah. And this is seen in 8% in male and 0.4% in females. So it is very commonly seen in males, but as compared to females, this is only like 0.4% in female, very less. Why? Because this gene is present on X chromosome. Okay. Son of a woman, son of a woman who carries gene has 50% chance of color blindness. The X chromosome coming from woman, like XY, this is son, and the if the X trait is coming from X chromosome is coming from female, then there is 50% chance of color blindness. Okay, yeah. For daughter to be colorblind, mother is carrier and father is colorblind. Okay, if daughter is colorblind, then it is like very sure that his father is also colorblind, because female contain XX chromosome and X is coming from father, and female is like carrier for that. Yeah. Now see, um, if we see X chromosome, if mother is carrier and father is XY, so X and XC, normal daughter, normal son, carrier daughter, and affected male will be formed in color blindness. Wow, we are just going to complete it. Yeah. Now, second is hemophilia. Hemophilia, with the name only, I think it's related to hemoglobinia. So, it is also sex-linked recessive disease. Single protein that is part of the cascade of protein involved in the clothing of blood is affected. Okay, one single protein that is a part of like chain of proteins involved in clotting of blood is affected. One protein is affected in hemophilia. Okay, simple cut, non-stop bleeding. If there is a simple cut also, then non-stop bleeding is being. Heterozygous female who is their carrier, okay, may transmit disease to son. Heterozygous female means heterozygous female, okay, may carrier. Heterozygous means the female which has carrier, means X and XH, okay, may transmit disease to son. And family of Queen Victoria as carrier of this disease. Queen Victoria was there na, in that also hemophilia was seen. So parent in, if female is carrier, then will be affected and daughter will be carrier daughter. Okay, sorry. Third is sickle cell anemia. Third is sickle cell anemia. This is autosomal recessive gene. See, all both color blindness and hemophilia was what they were, sex linked recessive trait disease. Okay, and this is autosomal recessive gene, means it is not linked with sex. Okay, both parents are carrier for the gene. Okay, in this both are there in both of hemophilia and colorblind. Female was carrier, and yeah, both parent carrier. Okay, and heterozygous. Okay, HBA and HBS. Okay, HBA normal, HBS carrier, and HBS deceased. Mutant hemoglobin molecule undergo polymer under low O2 tension. Mutant hemoglobin means alteration is there in hemoglobin, undergo polymer. This forms polymers like chains. Okay, under when there is low O2, low concentration of O2 in the cell. Okay, change of shape of RBC from biconcave disc to elongated like sickle. See, when there is सॉरी, आपको मैं पता नहीं क्यों एकदम से वो होने लग गया, बट सॉरी। See, when there is less concentration of O2 in the RBC, like we have read this in class 11th also in chapter breathing and exchange of gases. The same like phenomena is used in this. When there is less amount of oxygen is like in RBC. See, normally when O2 is okay, then RBC shape is biconcave disc. Okay, and when less concentration of O2 are there in RBC, then its shape changes to like elongated. RBC is this round, then this form elongated like sickle. Okay, that is why this named also sickle cell anemia. Okay, and the defect is caused by substitution of glutamic acid by valenic. So, because of the substitution of glutamic acid by valenic. Okay, so glutamic acid is removed and valine come place of it. That is why substitution is there. So sixth position of beta globin, sixth position of, so like hemoglobin structure is also there, like beta, para, meta places are there na. So same in like these are like above studies I have, but I have read in this in like my 11th class breathing and exchange wala chapter में ही. Sorry I speak Hindi, but like beta globin chain of hemoglobin single base substitution at sixth codon form GAG to GUG. Okay, yeah. Now we have read phenylketonuria, where we have read, just learn. I think in mutation, let me go back. Oh, no. I think we have read in this pleiotropy. Yeah, we have read in pleiotropy. Pleiotropy single gene and many characters. In bone error of metabolism, okay. Autosomal means not linked with sex. Affected individual lacks enzymes. The individual which is having this phenylketonuria have like have the lack is lacking some of the enzymes, okay, that convert the amino acid into phenylamine into tyrosine. So this is, I think this is also like somewhat based on breathing, because their amino acid is phenyl, no, there was pyruvate acid, yeah, I'm sorry. So the amino acid convert amino acid to like phenylalanine into tyrosine. Okay, so phenylalanine is accumulated and convert into phenyl pyruvic acid. Yeah, accumulation in brain cause mental retardation. Okay, excreted through urine because of its poor absorption by kidney. So why this is inborn meta, it is inborn error, autosomal recessive trait, amino. So in this phenylalanine is accumulated and convert into phenyl pyruvic acid, and because of this accumulation in brain cause, because of its accumulation brain cause mental retardation. Okay, so excreted through urine, and this is also excreted through urine because of its poor absorption by kidney. Okay, next is thalassemia. We have read color blindness, hemophilia, then third was here autosomal recessive was there, I think, phenyl sickle cell anemia, phenylketonuria, and third, fifth, sorry, one, two, three, four, five, yeah, fifth is thalassemia, autosome linked recessive. Okay, this is also autosomal recessive trait. Blood disease, this is blood disease. Okay, transmitted from unaffected carrier. This is transmitted from unaffected carrier. Okay, heterozygous means he or she is carrier, but he or she is unaffected from that carrier. Understood? So that is unaffected carrier. Heterozygous parent to the offspring. Okay, mutation or deletion result in a reduced rate of synthesis of one of the globin chain, alpha and beta. This is very important, and this is like tough to understand thalassemia. I'm just, but I will do today. Mutation or deletion result in reduced rate of synthesis of one of the globin chain, alpha and beta, that make up hemoglobin. Okay, abnormal hemoglobin result into anemia. Okay, alpha thalassemia. It is controlled by two. Wait, mutation or deletion result in what? Reduced rate of synthesis of one of globin chain, alpha and beta. Okay, that makes up hemoglobin. Alpha and beta are like making hemoglobin, and because of mutation this result in the rate of synthesis, means their making process is reduced, of whose? Alpha or beta, who are making hemoglobin. So abnormal hemoglobin result in an anemia. Okay, means hemoglobin is only will not be like processed, so anemia will be. So one is alpha thalassemia and one is beta thalassemia. Alpha thalassemia, it is controlled by closely linked genes. Okay, HBA1 and HBA2 on chromosome 16. Mutation or deletion of one gene or more of four genes, less alpha globin molecule produced. Okay, controlled by a single HBB gene on chromosome 2, on chromosome 11. Beta thalassemia, chromosome 11. Alpha, chromosome 16. Single gene, two genes. HBA1, HBA2, and HBB gene, only one. Okay, thalassemia is quantitative problem. Okay, quantitative means quality of hemoglobin is decreased, and sickle cell anemia is qualitatively. Qualitatively means quality. Quanti Thalassemia is quantitative means quantity is decreased, means hemoglobin are not forming, so quantitative. And sickle cell anemia is qualitatively means quality of mm, RBC are reduced, they have been sickle like elongated. Okay, HBA, HB2 chromosome mutation or deletion of one gene more than four, less alpha globin molecule produced. See, we have written alpha thalassemia में less alpha globin molecule produced, so quantity may decrease will come. That's right. Now next is last topic and very important. I'm very happy. Now next is chromosomal disorder, like disorders because of chromosome. These disorders were Mendelian disorder. Because of Mendelian disorders, Mendelian laws, and according to Mendelian, okay, Mendel, okay, and these are chromosomal disorder because of chromosomes. It is caused due to absence or excess of abnormal arrangement of one or more chromosome. Okay, failure of segregation of chromatid during cell division cycle result in gain or loss of a chromosome called aneuploidy. When there is segregation, process of segregation during cell division, see, division is there. Now these two chromosomes have to like divide, but due to its failure, they do not divide, so they gain or they completely lose. Yeah, so this one chromosome gained, but this organism completely lost. So failure of segregation of chromatid during cell division cycle result in gain or loss of a chromosome. This is only known as aneuploidy. What is it is called? Aneuploidy. 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 Example, Down and Turner syndrome. Okay, example, Down and Turner syndrome. bhai, this is very important. This will read in last topic, Down syndrome. Failure of cytokinesis after telophase stage result in increase in whole set of chromosome, polyploidy in plants. Okay. In according to, like, when we'll read cell cycle cell division chapter in class 11th, we have read there are many stages of cell division, like metaphase, telophase, S phase. uh, there are many phase. I have just like, I'll remind those later, but one of them is telophase, the last phase. Okay, so when there is in telophase, basically karyokinesis is done and cytokinesis is done, basically cytokinesis moreover. But when there is no cytokinesis, means there is failure of cytokinesis, means there is increase in set of chromosomes. Yeah, so if there is increase in set chromosome, that is known as polyploidy, and this is basically seen in plants. Okay. Human cell is basically having 46 chromosome, means 23 pairs, and 22 pairs autosomes and one pair of sex chromosome. Yeah, additional copy of chromosome is included when any other chromosome additional copy came, then it is trisomy. Okay, trisomy. And the lack of any, if, see, if these are there, it will gain trisomy, and it will, and this is losing. Yeah, this organism is losing this chromosome, means this is monosomy. Understood? Yeah. Now, on the basis of this only, lose and gain of chromosome, there are three syndromes. Okay. First is Turner syndrome. What is Turner syndrome? Absence of one of X chromosome. Okay. Karyotype 45, 44 plus O, means XO. Okay. This is basically seen in females. When they lack, they have two XX, but male have only X. So not in male, but in female, because it have two X chromosome, and absence of one of X chromosome is said to be Turner syndrome. So 44 plus X plus O. O means there is no chromosome, zero. Such females are sterile. Okay, and as ovaries are rudimentary. Ovaries are not forming any like hormones or any like organisms, so they are rudimentary, no work. So lack of other secondary sexual characters also. So they lack. Okay. Now next is Klinefelter syndrome. Klinefelter, like, see, pronunciation difference comes across many places. Like, I'm in my class, someone told, like, when I'm taking any lecture, they're calling it different. But according to me, it's not about how we pronounce it, it's about how we learn it and how we understand it. Okay, so yeah, Kline, Klein Felter, Felter, Klinefelter's syndrome, presence of additional copy of X chromosome. So in this lack was there of X chromosome, in this X is like gain, we have gained. Okay, presence of additional copy of X chromosome, karyotype 47 total, 46 are there, 47, 44 plus X plus X plus Y. Understood? Such individual has overall muscular development. Okay, feminine development, also development of breast and gynecomastia. Okay, such individual are sterile. Okay, such individual are also sterile. Okay. And Down syndrome, additional copy of chromosome 20, number 21, trisomy of 21. Trisomy was additional copy of any chromosome na. They are adding X chromosome, but the number of autosomal, yeah, 21 trisomal. This disorder was first described by Langdon Down. Down syndrome, Langdon Down. Okay, 1866. So Mendelian was still 1856 to 1863, and he have proposed 1866. The affected individuals short. The affected individual is how short. Okay, small round head. Okay, furred tongue, furred. Okay, and partially open mouth. Palm is broad, okay, and characterized by palm crease. Like, okay, physical and physiomotor and mental development is retarded. Means Down syndrome, short-heighted, open mouth, tongue frayed, round shape. Okay, mental retardation, physical, psycho and psychomotor retardation. Oh, palm are broad and their crease. Okay, these are. So our chapter is finished. We have realized we have read genetics, variation, inheritance, true breeding line, character, contrasting trait, inheritance of one gene, no blending, monohybrid cross, dihybrid cross, test cross, law of inheritance, law of dominance, law of segregation, incomplete dominance, codominance, inheritance, dihybrid cross, independent assortment, rediscovery of Mendelian work, chromosomal theory of inheritance, experimental verification of chromosomal theory, linkage and recombination, the theory, the words, the statements given by Alfred Sturtevant, polygenic inheritance, polyotropy, sex determination, its types, sex determination of honeybee, mutation, pedigree analysis, Mendelian disorder, color blindness, hemophilia, sickle cell anemia, phenylketonuria, thalassemia, chromosomal disorder, Turner syndrome, Klinefelter syndrome, and Down syndrome. Done. Now, according to me, we should take a rest, and today only, at this time only, we'll take rest. Okay, and we'll today only complete the NCRT of this chapter. Okay, and we, if we haven't got anything, then we come to the same video and we'll revise to that. Promise? Yeah. So this chapter is done, and hope you liked it. I know there are so many distractions in between, but according to me, this was like, I understand much better before, because I'm able to like make you all understand, na? So that's the main point, and that also in English. So we'll read this chapter, we'll take break, and we'll read this chapter. If any query, come to this same video. And thank you for watching this video. Stay tuned.